Welcome, everyone. Today, we are diving into the fascinating world of molecular phylogeny. Imagine trying to figure out how everyone at a massive family reunion is related to one another. You might look at physical traits, like nose shape or eye color. Molecular phylogeny does exactly this, but instead of looking at physical features, it looks at the very blueprint of life, DNA and proteins. By the end of this lecture, you will understand how we can read the history of life written directly in our genetic code. Now that we know what we are studying, let us look at how it works. In computational biology, molecular phylogeny is the scientific discipline of inferring evolutionary relationships among various biological species. Instead of relying on physical characteristics, which can sometimes be misleading due to convergent evolution, we use molecular sequencing data, such as DNA, RNA, or amino acid sequences. By aligning these sequences and measuring the statistical differences between them, computational algorithms can reconstruct the historical pathways of evolution. The fundamental assumption here is quite intuitive. Species that share a more recent common ancestor will naturally have more similar genetic sequences than species that diverged a much longer time ago. Understanding what molecular phylogeny is naturally leads us to ask why it is so important. Building these evolutionary trees is absolutely critical in modern medicine, agriculture, and conservation biology. For example, when a new virus emerges, scientists immediately use molecular phylogeny to trace its origins, figure out which animal it came from, and track how it mutates over time. It also helps pharmaceutical companies discover new drugs by identifying plants or microbes that are evolutionarily related to known medicinal species. Without this powerful computational tool, we would be completely blind to the evolutionary history that shapes the biological world around us. Before we can build an evolutionary tree, we need a way to measure how different two sequences are. This brings us to the simple p-distance formula. Conceptually, this formula calculates the ratio of genetic differences to the total sequence length. In this equation, p represents the p-distance, which is simply the fraction or percentage of genetic difference. The symbol n sub d stands for the number of mismatched letters, such as nucleotides or amino acids, between the two aligned sequences. Finally, n represents the total length of the sequences being compared. Physically, if the number of mutations increases, the distance p also increases, which tells us that the species are less closely related. Now that we have our formula, let us see this with a concrete example. Suppose we are working in a genetics lab and have isolated two short DNA sequences from two different frog species. Sequence A is ATGCGT, and sequence B is ATGGGT. First, we align them and count the total letters, giving us a denominator n equal to 6. Comparing them letter by letter, we find only the fourth letter is different, meaning our mismatches, n sub d, equals 1. Dividing 1 by 6 gives us approximately 0.167. This physically means there is a 16.7% genetic difference between the two frog species for this gene segment. Having calculated the simple genetic distance, we must now address a major limitation. The simple p-distance actually underestimates the true evolutionary distance. Why? Because over millions of years, multiple mutations can happen at the exact same genetic spot. For instance, a nucleotide might mutate from A to T, and then later from T to G. When we look at it today, we only see one difference, completely missing the intermediate step. These are called hidden overlapping mutations. Because of this, the raw percentage of differences we observe on our computer screen is lower than the actual number of evolutionary changes that took place. Therefore, we need a mathematical model to correct for these hidden mutations. To solve the problem of hidden mutations we just discussed, we use the Jukes-Cantor model. This formula mathematically stretches the observed distance to account for multiple substitutions at the same site. Here, the variable d represents the true evolutionary distance. The variable p is the simple p distance we learned to calculate earlier. The natural logarithm function, denoted by ln, is what mathematically stretches the distance as p gets larger. Physically, when the observed difference p increases, the corrected distance d increases exponentially. This reflects the high probability that more and more hidden, overlapping mutations have occurred over long periods of evolutionary time. Let us apply this correction formula to a real scenario. Imagine we are comparing two highly divergent bacterial species. Out of 100 total nucleotides, exactly 30 are different. First, we find the simple p distance, which is 30 divided by 100, giving 0.30. Next, we substitute this raw value into the Jukes-Cantor equation. We multiply 0.30 by 4/3, giving 0.40, and subtract that from 1, leaving 0.60 inside the parentheses. Taking the natural logarithm of 0.60 yields approximately negative 0.51. Multiplying this by negative 0.75 gives a final corrected distance of 0.383. This physically tells us the true evolutionary distance is 38.3%, revealing that about 8.3% of mutations were hidden. Now that we have calculated these genetic distances, let us visualize what they actually build. You are looking at a simple phylogenetic tree diagram. On the right side, you see the tips of the branches, labeled species A, species B, and species C. These represent the organisms as they exist today. Notice how species A and species B join together at a node relatively close to the present day. This indicates they share a very recent common ancestor. If we follow their shared branch further back to the left, it eventually connects to the branch for species C at the root of the tree. The horizontal axis at the bottom represents evolutionary time, or genetic distance. The longer the horizontal branches, the more genetic changes have occurred. Having seen what a phylogenetic tree looks like, we must now ask, how do we actually draw it from our distance calculations? This is where UPGMA comes in. UPGMA stands for Unweighted Pair Group Method with Arithmetic Mean. It is a straightforward, distance-based, agglomerative hierarchical clustering algorithm. Imagine you have a room full of strangers and want to group them by age. You would find the two people closest in age, pair them up, treat them as a single unit, and then find the next closest person. UPGMA works exactly like this. It constantly hunts for the most similar pairs of species in a distance matrix and groups them together step by step, assuming a constant rate of evolution across all branches. To perform this step-by-step grouping, we need a way to recalculate distances every time we form a new group. This is the UPGMA average distance formula. Conceptually, when we merge two closely related species into a single cluster, we need to know how far this new cluster is from all the other remaining species. In this formula, d sub ij, k represents the new calculated distance between the newly formed cluster, made of species I and j, and another existing species k. The variables d sub ik and d sub jk are the original genetic distances from the individual species to species K. The formula simply adds these two original distances together and divides by 2 to find the arithmetic average. Physically, it represents the average evolutionary divergence of the new group. Let us put this algorithm into practice with a 3-species example. We have species A, B, and C, and we have already calculated their pairwise genetic distances. The distance between A and B is 4, A and C is 8, and B and C is 10. The very first step of UPGMA is to look at all available distances and find the absolute smallest value. Here, the minimum is 4, which is the distance between A and B. Because this is the smallest value, we merge species A and B into a new cluster called AB. Since they are separated by a total evolutionary distance of 4, the branch length from their shared common ancestor to each individual species will be exactly half of that. Therefore, the branches leading to A and B will each have a length of 2. Now that we have formed cluster AB, we must update our distance matrix. We need to find the distance from our newly formed cluster AB to the remaining species, C. We take the original distance from A to C, which is 8, and the original distance from B to C, which is 10. We add these two values together to get a total sum of 18. Finally, we divide this sum by 2 to find the arithmetic mean, as dictated by the UPGMA formula. The new calculated average distance is exactly 9. This physically means that the average genetic distance from the AB ancestor group to species C is 9 units of evolutionary time. We have successfully reduced our complex 3-species problem into a simpler 2-group problem. We're now ready for the final step to complete our tree. Continuing directly from our previous calculation, we have only 2 entities left, the combined cluster AB and the single species C. We just calculated that the genetic distance between them is exactly 9. Since there are only 2 groups left, the algorithm dictates that we simply merge them together into the final root of the entire tree. To find the branch lengths from this ultimate root, we take the distance between the 2 groups and divide it by 2. Dividing 9 by 2 gives us 4.5. This physically means that the total evolutionary time from the very first common ancestor of all 3 species down to species C is 4.5 units. Our tree is now completely resolved and mathematically scaled. We have covered a lot of ground today, so let us summarize the most important ideas. First, molecular phylogeny allows us to read the history of life written directly in our DNA and protein sequences, providing a much more accurate picture than physical traits alone. Second, we learned that simple genetic distances can be misleading, and we must use models like Jukes-Cantor to correct for hidden, overlapping mutations that occur over millions of years. Finally, we saw how the UPGMA algorithm uses these mathematical distances as foundational building blocks to construct clear, hierarchical maps of evolutionary relationships from the bottom up. You now have a solid foundation for exploring more advanced tree building methods in bioinformatics. Great job today.